Hey, PBio. So we're going to take a look at the second group of free response questions, and this one is one of my favorites. I really like this because they outline the three major reactions, actually the four, they combine one of them into two, of cell respiration. And so we have three figures here. Figure 1 showing glycolysis and pyruvate oxidation, Figure 2 shows Krebs, and Figure 3 shows the electron transport chain. And again, what's going on here I just want to pause a bit and just kind of recap. We're taking glucose. Notice you've got 6 carbons in the glucose molecule, right? And we're taking this glucose, we're hydrolyzing it down into pyruvate. Okay, we're clearly shown what's being released here: 2 ATP, 2 nada, okay, and 2 pyruvate. And then we're taking that pyruvate and we're breaking it down even further into acetyl-CoA. Okay, and what we can see here is that we've got 6 carbons being broken down into 2 3-carbon compounds, and then these are broken down into 2 2-carbon compounds. Okay, and we release the CO2 as waste. And then from here, we move into the Krebs cycle. So here's our 2-acetyl-CoA going into Krebs. And we can see all the stuff coming off of Krebs. Okay, there's NADA, FADA, GTP. What the heck is GTP? Pause, relax. The same thing as ATP. Okay, energy being released. But also notice on Krebs that we're getting rid of CO2 as well. Okay, so we're completely breaking down the 2-acetyl-CoAs. So 2-acetyl-CoAs have 4 carbons in each, and here are 4 carbons coming off. Okay, and then from there, this NADA and FADA goes into our electron transport chain. So we can see that NADA is dropping off hydrogens, right? And that these hydrogens are being split into protons and electrons, okay? And it's pretty clear to see that these protons and electrons, when they get split, protons go on one side of the membrane, and here are electrons moving through the electron transport chain. Okay, so what do they ask? Okay, first they give you the little blurb about cell respiration and all the processes, and they keep it pretty vague. Now they want you to use the information above and describe one contribution of each of the following in ATP synthesis. So, catabolizing glucose in glycolysis and pyruvate oxidation. What does that contribute to ATP synthesis? What does breaking down the intermediates, oxidation of the intermediates in Krebs, contribute to making ATP? And how does forming a proton gradient by electron transport contribute to ATP? So here's what they suggested. It's quite a bit here, right? Glucose and glycolysis, we know that we're going to produce NADA. We're producing the acetyl-CoA for the Krebs cycle. So we're contributing the hydrogens in NADA for electron transport. The acetyl-CoA is important for getting that Krebs cycle going. We also make ATP because this is going to allow us to phosphorylate ADP to ATP. Okay, Krebs cycle. Number one thing it does is it produces all that NADA and FADA for the electron transport chain. It also allows us to have that high energy electron that we need for the electron transport chain. NADA and FADA provide the energy to pump protons against their gradient. Okay, and then they even threw in here a little GTP, right, for substrate level phosphorylation of ADP. And then lastly, that proton gradient, the whole point of that is to generate ATP, right? Having those protons move through ATP synthase allows for the production of ATP, and it also provides energy for oxidative phosphorylation of ADP to ATP. Okay, not bad there. Okay, second part. It says, use each of the following to justify the claim that glycolysis first occurred in a common ancestor. Okay, so we know that glycolysis happens in all existing organisms, right? And this shows that it was an advantage, and that gene or whatever the process, the traits, they formed early on, and they were passed on. We know glycolysis happens under anaerobic conditions because glycolysis happens before there was oxygen. So it occurred in early Earth conditions when there was no oxygen. And we know that glycolysis occurs in the cytosol, and that's because the origin of glycolysis, again, happens before we see eukaryotes. So glycolysis was first started in these early prokaryotic cells. Okay, and then we have letter C here. Love this one. So this is where you were given the energy that is in the total oxidation of glucose. And we know that if you hydrolyze glucose and oxidize it completely, you get 686 kilocalories per mole. We know that 1 molecule of ATP, you're getting 7.3 kilocalories per mole from 1 molecule of ATP. So the first thing they want you to do is say, okay, if 1 molecule of ATP gives you 7.3 kilocalories per mole. How many kilocalories, how much energy would you get from 30 moles of ATP? So 30 moles of ATP is 30 molecules of ATP. So all you do is multiply 30 times 7.3, okay? And that gives you, let's see if I can do this without revealing too much, 219 kilocalories, because all you're doing is taking 30 and multiplying that by 7.3. The second part of this asks you to calculate the total efficiency of AT production from glucose in this organism. So we know that glucose is making 30 moles of ATP. So we're making 219 kilocalories. There are 686 kilocalories per mole in a complete molecule of glucose. So what this is asking is what percent of the energy of a glucose molecule goes into making ATP? And the answer to that is about 32%. Okay, what we're doing is we're taking 219, okay, which we got here, and we're dividing that by 686. So out of the total amount of energy available in glucose, which is 686 kilocalories, 219 kilocalories go to formation of ATP. So that comes out to about 31 to 32% of the total energy available in glucose. So the big question is, where does that extra energy go? Yep, it's heat, right? It's released as heat energy or as waste. Yeah, that's it. The other thing is you can even say that some of that energy is in the form of waste products like CO2 and water. Okay, that's legit as well. Okay, and then finally, the last part here says that the enzymes of the Krebs cycle function in the cytosol of bacteria, but among eukaryotes, they're found mostly in the mitochondria. Ah, there we go. So they want you to pose a question that connects the subcellular location of the enzymes in the Krebs cycle to eukaryotic evolution. This relates directly to the endosymbiont theory. Okay, we know that Krebs cycle happens in the cytosol of bacteria. It happens in the mitochondria in the matrix. So we know that the matrix of the mitochondria is similar in its environment and chemical makeup to the cytosol of bacteria. So they want you to ask a question. Okay. So something like this could be, since the Krebs cycle occurs in the cytoplasm, does it suggest that the mitochondria were once prokaryotes? Boom, done. Okay. Does this, another scientific question related to evolution, does this suggest that mitochondria eventually evolved into that prokaryotic cells eventually evolved into mitochondria? Does this show that mitochondria were free living on their own at one time? Okay. And that's all you needed to add for that. Okay. So that's question 3. I'm sorry, question 3. That's topic 3, question 1B. That's that big question. And we'll jump back in a second. And question 2 of group B, we have this graph, which shows the initial rate of an enzyme catalyzed reaction at different substrate concentrations. And so they want you to first connect the primary structure of the enzyme to its overall shape. So we know that the amino acid sequence determines the shape of the protein. So what you can say for your response is that the amino acid sequence determines the shape. The R groups on the side chains interact and they stabilize the structure and they help maintain the shape of the enzyme. Okay. Number 2, they said, or letter B, they said predict the effect of adding a non-competitive inhibitor to the reaction mixture on the rate of reaction at a high substrate concentration. So you can support your prediction by describing how it affects the structure and function of an enzyme. So a non-competitive inhibitor will decrease the initial rate of the reaction. And the way it does this is it binds to someplace else besides the active site. So it's going to bind to a regulatory site or an allosteric site, and that's going to alter the interaction of the substrate, and it's going to lower the rate of reaction. Okay. And then for letter C, provide an explanation for the shape of the curve enclosed in area 1 and how it changes in area 2. So in area 1, the enzymes are interacting with the substrate. They are increasing the rate of reaction because more enzyme is working with more substrate, and the rate of the reaction increases. And in area 2, the enzymes are interacting with the substrate, and they are completely saturated. So they are operating at Vmax. Okay, at this point here in area 2, they're almost completely saturated, and then eventually it levels off because they are working at their maximum capacity. We have a limited amount of enzyme working with an unlimited amount of substrate. So there's a point where all the enzymes are occupied with substrate, they can't work any faster. They're working at their maximum capacity. Okay, so those are your answers for Group B on topic 3. I hope that helps. And again, if you have any questions, anything you're not sure of, please reach out to us and let us know. Okay, we'll talk to you soon. Bye, guys.